You hear her?
No.
On there now, broadcasting. Hi, this is Fred Gage. I'm your lecturer for today. I'd like you to read this CME continuing education disclosure. And at the end of the talk, or during the talk, you can post some questions in the question and answer, write them down, and I'll try to get to most of them as we go along, or at the end of the talk. I'll put some time into answering some of your questions. Okay, so if you've read all that, I'll move on to my first slide of the presentation, which gives the title, Function and Regulation of Adult Neurogenesis. And I'll be speaking today. The whole idea of adult neurogenesis came somewhat of a surprise for people, and that we were taught early on that the brain was fixed and the number of cells that we have in our brain remains constant throughout life. And there are two critical issues here. One is that if cells were dividing all the time in the brain, we'd have no consistency, wouldn't be able to keep a memory. And the other is who we are as a person. The second point really was that how could a neuron actually undergo cell division? It's such a complex structure. So, and we know that these neurons here, and I'm going to use my tool to point out for you things. So here I have my tool. You see how neurons are really complex structures. They have branches. The idea that this cell could undergo cell division was unusual, or let's say at least. And we have 10 billion neurons in the brain and hundreds of thousands of connections. So the idea that the cells could undergo cell division was odd. Well, this was resolved by the fact that two things. One is that neurogenesis, or the birth of new neurons, did not occur in every area of the brain, but was restricted to just a few areas of the brain. So the whole brain wasn't turning over all the time. And the other point is that cells really didn't divide. Neurons didn't divide, but in fact there were stem cells, immature cells existing in the brain, and those are the ones that gave rise to absolutely new neurons. So those were, with those new facts coming along, we were able to accommodate these concepts. And there's two areas of the mammalian brain where neurogenesis occurs. One is in the lateral ventricle region. Let's see if I get my pointer correct. This region right here. And the cells that are born in this area here then migrate along this pathway here where they reach the olfactory bulb. And once they arrive in the olfactory bulb, they pass through the granular cell layer, or giving rise to some cells here, but also periodically giving rise to neurons, but bypassing these mitral areas in there. I'm just learning how to do this pointer, so bear with me here. The other area of the brain where this occurs is a structure called hippocampus. And hippocampus is the structure that is involved in learning and memory. And the cells are born right in here. It's called the subgranular zone. And this is where the stem cells proliferate and then migrate and eventually differentiate. And I'll go into this in somewhat more detail. So this is a real picture, not a cartoon, and it shows the area specifically of the dentate gyrus where these new neurons are being born. They're represented by these green cells, and I'll tell you how they're labeled in a minute. All the cells in the dentate are, for the most part, excitatory neurons. Inside here we have inhibitory neurons. And interestingly, the cells that are born in the subventricular zone and go to the olfactory bulb, they're inhibitory. So there really is a difference in the fate, at least, of the stem cells that are born in these two different areas. So a little more detail about anatomy. Here's the whole hippocampus again. Here's our dentate gyrus. This is called the CA3. This is where the fibers from the dentate project. And CA1 is where CA3 then projects. And The inner zone between the two leaves of the dentate gyrus is called the hilus. We'll go into that in more detail. And this molecular layer here is where the branches from the granular cells move into. So this is what we call the niche. Here's the dentate to get you oriented again. These are the stem cells. They're surrounded by blood vessels and astrocytes, keeping these stem cells nourished. They have to move away from that niche in a few days to get rise to their neurons with branches. All of it takes about a month to 6 days before the immature cell is fully mature, more like 6 to 8 weeks. Here's the whole structure now, giving us an idea of the dentate granular neurons, with the inner cells here being the hilus cells, the inhibitory neurons that are suppressing the activity of these excitatory neurons. These new cells sending their processes out to the CA3, the CA3 then going to the CA1 and then back out again to the entorhinal cortex. It's the entorhinal cortex E2 that gives us these cells here that then go all the way to the dentate granular neurons. They go from here to there, there to there, and then back out again. So that's the synaptic circuit of the hippocampus, how information is processed in the brain. And new memories are formed. So here's that diagram again, and what you see here is that there is the niche is here. This is where the cells are born. And here we can see down here that there are various transmitters that regulate the fate of the cells at various stages. So we have progenitor cells that go to differentiated cells, all the way up to the mature cells. And you see that GABA is controlling at various stages of this process, from stem cells on down. Glutamate and acetylcholine are more involved in the process of maturation, and the aminergic system is controlling them at earlier stages of differentiation. We should also, and I'll talk about this, give you some details about this in a little while, but the rate that neurogenesis occurs and the number of cells that are born is influenced by activity. So physical exercise like running can increase neurogenesis, and enrichment can support survival, so you end up with more new cells, while stress is a negative regulator and decreases the number of proliferating cells. Again, the circuitry showing this inputs from the entorhinal cortex, again, to the hippocampus, from the neuronal cortex to the, this is actually a good one, I'm still learning a little bit here. Yeah. From neuronal cortex to dentate, dentate to CA3, CA1, and then back out again. Now we get a little more detail about the timing of this. We think that there are these very primitive cells that are non-dividing for the most part. They can give rise to a rapidly dividing cell that's after this division occurs, this initial division occurs. It takes about 7 days for the first processes to get out, and then they don't get their inputs till later. I'll be repeating this with various examples over time. One thing is clear is we now know a lot about the molecular markers that identify the cells at various stages. So the quiescent, mostly quiescent stem cells are GFAP positive, SOX2 positive, and nestin positive, whereas the proliferating population of cells are SOX2 positive, nestin positive, and TLX positive, but they're not GFAP positive. These cells here are the stem cells, or these are the quiescent stem cells, these are the rapidly dividing cells, and these are the neurons. Neurons in the hippocampus express a variety of markers that identify them as being mature, in particular PROX1 and NUN. Variety of ways to mark these cells as being newly born. We can use a nucleotide analog, nucleic acid analog called bromodeoxyuridine, which substitutes for uridine when the cell is undergoing cell division, so it incorporates into the DNA after a single injection. So all these black dots correspond to newly born cells. In other cases, we use transgenic animals that express markers of the cells at various stages. In addition, we use retroviruses. Retroviruses are viruses that can be modified so they're non-toxic, but express a gene like GFP that can mark the cell that undergoes cell division, and the cells stay marked. The advantage here is it fills up the cytoplasm. So here's an example of the fact that we can see this neurogenesis now in every mammalian organism that has been identified so far. So from mouse to human, and the dentate itself looks very much the same in all these structures. They all have PROX1. They can, here in this human study back in 1998, patients that had cancer were administered bromodeoxyuridine to see how fast their tumor was growing, and subsequently, when their brains were examined, it was shown that there was an incorporation of BRDU into stem cells that became neurons and survived for years in their brains, evidencing for the first time that there was adult neurogenesis in humans. This was subsequently confirmed years later, many years later, in a Swedish study by Spalding and Friesen, showing more quantitatively that up to 800 new neurons are being born in the human hippocampus on a daily basis. So here's what some of these retroviral injections look like and how they function. This is a retrovirus, has three components: the virus itself with core proteins and a pseudo envelope that allows it to infect mammalian cells easily. In stereotaxis, we inject this virus into the brain, where it is integrates into just those cells that are undergoing cell division within the structure here. And the advantage of it is that it fills up the cell and the cytoplasm, not just the nucleus. So here at 3 days, you can see the cells that have just been infected, and they're very immature, not many processes. By 7 days, they have already begun to show processes here. And then by 10 days, you have long processes going up here. And by 14 days, they branched a little bit more even. And then by 21 days, it's even further branching. And importantly here, we are all the way out to 14 months after the initial injection, the cells survive and are fully integrated into the circuit. Here's a study asking the nature of these cells. So can we use one of these markers like SOX2, which identifies the stem cells, and be certain that they really are coming from this SOX2 positive cell? So we've used a variety of molecular techniques to do this. Here we have a transgenic mouse expressing SOX2 off the expressing GFP off the SOX2 promoter. And you can see here that the brain is GFP positive. If we now look inside the brain, we can see that the dentate gyrus is GFP positive. So it seems to have worked. So our SOX2 positive cells are there, but we need to confirm this. And so you can see that when we double label with some of our other markers, we have SOX2 positive cells with long processes, likely the immature radial cells, and also we have some of these small non-radial cells is there as well. So SOX2 is labeling both the quiescent cell as well as the mature cell. The advantage of this mouse is that we can actually sort for GFP and pull down this fraction of primitive cells that exist in there, about 6% of the total population of dentate cells. They can be propagated in culture. There's SOX2 positive, nestin positive, GFP positive. Negative as I showed you that the proliferating cells are GFAP negative, whereas the quiescent ones are GFAP positive. So here we're proliferating the we call type 2 or the proliferating neurostem cells. These cells then can be induced to differentiate into neurons, TUJ1 positive, double cordon, as well as oligodendrocytes and GFAP positives, evidencing the fact that these really are stem cells since they can be isolated, propagated into different cell types. Now, to do fate marking, what we've done is to take a lentivirus that has a SOX2 promoter driving GFP/CRE, and we inject that into a floxed mouse, floxed negative mouse. So when we knock out that flox with the CRE, then we get a recombination event occurring and the cells turn green. So we can mark cells permanently that had been SOX2 at the time of the injection of the virus. Then we can pulse with BRDU and follow the cells at various time points later to see whether or not what the fate of that SOX2 positive cell was. And when we do that, we can see that here's a SOX2 positive cell, or was at one point, and some 28 days later, it had undergone cell division at least once, and it's still SOX2. So that means that some of the stem cells are dividing and retained within the subgranular zone. Others can give rise to neurons. So here's a SOX2 infected cell. So it was SOX2. It underwent division, it BRD labeled, and now it's double cordon positive. And here's another one that's PROX1. So they have capacity for giving rise to multiple lineages. And here's the GFAP positive cell. This is rare. They don't give rise to GFAP SOX2, or sorry, GFP SOX100 cells very often. Usually they give rise, most of them give rise to neurons. So the conclusion here is we think that this proliferating cell is the one that gives rise to neurons in glial cells, and there is some evidence that this can also de-differentiate into a quiescent cell. We think of these as the initial cell, but they're not giving rise directly to neurons, but rather it looks like they're giving rise to this intermediate stem cell that then gives rise to the other cell types. Okay, so let's look at what role the local environment might play. Is there something unique about these cells? Interestingly, when we pulse an animal with bromodeoxyuridine, we can find BRD-labeled cells all over the brain, in the cortex, and certainly in the subgranular zone, but also even in the white matter. So why don't we get neurons in all other places? So what we can do is we can isolate these stem cells directly from the brain and inject them into either a neurogenic region or a non-neurogenic region. So here we've taken stem cells directly from the brain and injected them into the hippocampus, and they can give rise to mature granular neurons. When we inject hippocampal neurons into the olfactory bulb, they can go all the way out to the olfactory bulb area and give rise to olfactory neurons, suggesting that these cells can behave according to the environment that they're in, so they're not restricted in terms of their capacity. If we inject them into the spinal cord, what we do, we see that they don't give rise to neurons because there are no stem cells giving rise to neurons there, but they do survive, migrate, and give rise to glial cells. So what is it about the environment that's so important for giving rise to neurons exclusively in the olfactory bulb and the hippocampus? This is a Nemarsky image of the Dentate gyrus, and you can see that there's this blood vessel, and these cells are in the cusp of the blood vessel, so they're getting some sort of nutriment from the blood vessel. So that seems to be one of the important features of this. This is actually a lower power showing the labeled cells, GFP green neurons here, and you can see the highly rich vascular nature of this. So blood vessels seem to be an important feature of this. In addition to blood vessels, astrocytes are important. So here we're labeling with BRDU and with GFAP, and we find that they're surrounded by blood vessels and with astrocytes, forming this niche for the cells. So let's see if there's something important about the astrocytes. When we take these cells out of the brain as cultures and plate them on hippocampal astrocytes, they will spontaneously differentiate into neurons, suggesting the astrocytes are making something that allows them to differentiate. If, however, we put them on astrocytes from spinal cords, they don't differentiate. So already we know that there's something unique in the environment that allows them to differentiate on hippocampal astrocytes, but not spinal cord astrocytes. So we've gone in to dissect out what was unique about these astrocytes in the two regions of the brain, spinal cord and hippocampus. We find that in the spinal cord, in the hippocampus astrocytes, they contain all the major pathways for the Wnt signaling. So the receptors all the way down through the TCF-left, which is the DNA binding domain for the canonical Wnt signaling. But what they are missing, these are the neuroprogenitor cells, have all the signaling pathways, so they can be affected by Wnt. What they don't have is any of the Wnt factors themselves, the ligands. But when we look into the astrocytes, we see that they are, in fact, contain Wnt activity. And when we test for the effect of blocking Wnt signaling, we can show that we can decrease the amount of neurons that are being made. So Wnt seems to be, in vitro, a crucial factor in taking the cells from an undifferentiated state into a differentiated state. We prove this very convincingly by using a dominant negative Wnt, so that's going to block Wnt signaling, and inject that into the hippocampus with a lentivirus, which blocks Wnt signaling. And then we look for a marker of neurogenesis, which would be double coordinate. Here's the control virus. You can see the red is all double coordinate. The cells are fine with the control virus, but it blocks neurogenesis in the dominant negative lentivirus. This is the quantification of that at this point. So what more do we know about this? We know that NeuroD is a transcription factor that's really important in the transition from a stem cell to a neuron. It's sort of the middle stage to a neuron. And Wnt signaling, it appears to be an activator of NeuroD. In fact, within the promoter of NeuroD, there are a number of these beta-catenin TCF-left binding sites. And it is through, we believe it's through this direct application, activation of this promoter that the canonical Wnt signaling system turns on the neural differentiation program. These are some studies showing that when you block NeuroD in vitro, you can block the differentiation of the cells into neurons in this DOX-regulated, controlled NeuroD cells. And here in vivo, in a study that was published by another group, showed that when you knock NeuroD out entirely, in this group here, we get a complete loss of the dentate gyrus, showing that NeuroD is obligatory for the formation. So we look at it in a more general way. We think that when signaling at early stages is involved in proliferation of the cells, but then it activates a initial cascade that induces the cells to differentiate. So it's a crucial factor through its activation of NeuroD. Interestingly, the canonical Wnt signaling pathway decreases as the cells differentiate, and the non-canonical pathway plays a role in dendritic arborization, a story that I won't be able to get to today, but was recently published in the Journal of Neuroscience. So all in all, we've got an interesting conundrum here. We've got a new bunch of cells coming in. We know that. We've proved that they become neurons, but they're receiving, well, they potentially can receive inputs from the neuronal cortex and then send out axons to the CA3 and also get interactions with these higher cells. The question is, in the adult brain, are they actually integrating fully into the system? Here is a cartoon. This is actually a real picture of a GFP labeled newly born neuron, and these little protrusions are called spines. Spines come in different flavors. They have these filopodia that when they're very young, and then they begin to mature, they become stubby, and then they're a little larger, and then they have these large mushroom bodies when they're non-mobile and quite mature. So we can quantify these in vivo and in vitro in these cells. And what we find is that the newly born cells don't have any spines when they're 14 days, but by 21 days they have a lot, and then they become very dense with spines later. A finer dissection shows that it's really the transition between day 15 and 16 that they get spines. By 17, they're on. So this timing of spine development is very, very specific. We've gone on to look very specifically whether or not the intraocular cortex, which is the input from the cortex to these granular cells, make contact. So here's a green GFP positive cell. Here, it's a green GFP positive cell. And we use a red virus in the intraocular cortex, which is a lentivirus, and it tracks the axons as they go from the intraocular cortex to the dendrites of these cells. So that's the red up here. And here you can see a red presynaptic terminal on a green postsynaptic terminal, suggesting that the intraocular cortex actually makes direct contact with these newly born cells. And here we've taken this to the electron microscopic level, so we can get photo bleaching of this green fluorescent dye so the cells turn black, the processes turn black. And here's a spine in a newly born cell making contact with the presynaptic terminal with all these nice little vesicles. In there. You see here's the presynaptic side here, and this is the dense core vesicles. This is the postsynaptic density here. In addition, on the other side, so these are the axons that are coming out of the axons coming out from the mossy fibers going up to CA3 region here. We look at this in higher power. We can see that there are, in fact, big mossy fiber terminals from a newly born cell making contact on the dendrites and on the shafts of the existing CA3 neurons. So all in all, we can see now that newly born cells make contact both with the inputs coming in from the neuronal cortex on their spines, and the newly born mossy fiber terminals can make contact with their partners on the CA3 region right here. We looked at the time course of this, and what looks like happening is that there's a temporal nature where the filopodia form first, and then later on we get to mature mossy fiber terminals. And it looks very much like there's some sort of competition that's going on. And we hypothesized that these fine processes here, filopodia here, compete with the existing synapses, and they get larger once they get there, and finally they compete out, and where this one is now lost, and the sole new one takes its place. So this is an example of that. Here's an example of that competition underway. And we see the same thing happening in the axons. So that over on this side here, you can see the inputs. This is the time course of this process of the axons moving into the CA3 field. So We have both a pre-synaptic and a post-synaptic competition. So we don't think there's actually new synapses being formed, too many, but rather the formation of competition of a pre-existing synapses that constitute this process. Here's a cartoon of that. So you have a pre-existing synapse. Here's the new Philippodia coming in. Over time, they have this direct competition, and one runs out. There are a bunch of theories, and other people are testing this hypothesis right now. One suggestion is that the astrocytes, which are not depicted in here, are playing some key role in regulating this process, but it remains to be seen. This is on the presynaptic side, where the new terminals coming in from the newly born cells are competing for this spine with the existing synapse. So we know now that they make anatomical connections. The question is, are they physiological? Well, the first evidence for physiological and electrophysiological activity of these newly born cells came in 2002. 2004, an interesting paper showed that the young cells, when they're moving in, have a different activity there. They seem to be hyperexcitable, and this was confirmed later on. So what is this hyperexcitability all about? So what I want you to look at here are some electrophysiological tracings that show that the immature cells, so here we'll look at 1 month of age, in the new, so the cells are 1 month of age in a mature neuron, and this regular LTP-like response is what I would call a long-term or hyperexcitable LTP, whereas in mature cells we have an LTP, but it's much lower level. So this is the hyperexcitability that's been reported by many investigators of the young cells that seems to go away. So people wanted to know what this hyperexcitability really is all about. This image didn't come across very well. I hope this is just an anomaly. But it does show that the 4-week-old animals have this hyperexcitability from baseline, whereas the 8-week-old don't have it in the presence, if you don't block inhibition of GABA. So there's so much GABA normally in the slice that inhibits things. If you block GABA, you can see the hyperexcitability. But interestingly, in the young animals, it doesn't matter if you block GABA or not, they're hyperexcitable. That gives you a hint of what I'm going to tell you about in a minute. And that is that the, this is also distorted a little bit, but that here's this hyperexcitability in the young cells. And if we give GABA, we can suppress that. However, in the old cells, we have to block GABA in order to be able to see the elevated amount of LTP. So GABA is playing two different roles in the young versus the old cells. So what's the source of the inhibition that we see? Actually, here's this hippocampus scan. We've got a lot of inputs. And I told you that these transmitter systems could regulate neurogenesis, and they all send axons into this area and regulate it. But what is the source of the GABA that we're seeing? We used a technique called rabies virus tracing, neural tracing with rabies virus. Rabies virus naturally propagates between neurons in a retrograde manner. And colleagues here at the Salk, John Young and Ian Wickersham in Ed Calloway's lab, developed this as a technique that we use for tracing neurons. And they use a modified rabies virus, which is normally toxic, so that it won't jump across more than one synapse. So it's restricted to only labeled cells that are connected by monosynaptic connectivity. And I'll show you an example of this. Here we have the model that we've used, and this is based on a transgenic animal made by Martin Goulding and Flora Stam, which has a flock stop which normally prevents the rabies glycoprotein from being expressed. And if you use a Cree retrovirus, which is only going to label the dividing cells, remember, we can flux this out and express this TVA. So now we can come back at a later time point with the virus, the rabies virus, which has a deleted glycoprotein, and inject it in whenever we want and see who's connected to the newly born cells right at that very moment. So it's a very effective way of selectively labeling just the newly born cells. And this was some work done in a postdoc in the lab, and here's just showing that it works. You can see the yellow or the double labeled cells, so they are the cells that were originally labeled, and the red ones without the yellow are the cells that are connected to those cells. So this is all within the dentate, so there's a lot of connections right there nearby. But if we look distal to it, here we are in the interrhinal cortex. Remember, that's layer 2, so those are the cells that connect to the newly born cells, and this is showing that they're connected, sort of a validation the technique works. Interestingly, in the medial septal area, we're getting nice connections of cholinergic and GABAergic neurons that are in the medial septal area that project up into newly born cells. So they're connected there. And we see in the midbrain cells that are labeled in the VTA as well. As expected, we see cells in the hilus. These are these large hilar mossy cells. These are, in fact, excitatory neurons in the hilus, as well as inhibitory neurons in the hilus as well. So we're getting a lot of connections. We had a little bit of surprise in the molecular layer. So this is out where the dendrites are. We found these large cells that had been identified by Thomas Freund and Yuri Rizhaki some years ago in the molecular layer, called MOP cells. And they are also connected to the newly born cells. This is a single cell, and this is all of its dendritic and axonal processes that are innervating there. So a single cell has an inhibitory cell, also has a control from this region. When we record from these cells, we see that they are, in fact, inhibitory cells in the slice. So the theory, and we think how this works, is that when a stimulus comes in from the neuronal cortex, you have some direct input to the granular cells, but there's also a feed-forward inhibition here, where some of the stimuli go through these inhibitory neurons to shut the cells off. And here you can see the timing of this. So this is an excitatory postsynaptic term of EPSP with short duration. That would be the direct. This would be the direct connection from the neuronal cortex here. And then you have this one here with the delay, and that is because you're recording from here and you're going through the inhibitory neurons before you get there, so you have a delay. So the first is an excitation, followed by an inhibition, regulating the duration of the response. So here we have potential for a feed-forward inhibition mechanism from the molecular layer, mediating this maturation-dependent inhibition of the newborn granular neurons. And here it just gives you a summary of these MOP cells that are called in the molecular layer, inhibitory neurons that we identified, and that the granular cells are receiving input from both of these locations. So the conclusion is that cortical and subcortical local inhibitory inputs are identified by genetic rabies, genetic viruses using rabies. During maturation, local inhibitory inputs to newborn neurons are gradually increasing. They become excitatory. This is during what we call the critical period. This developmental change in inhibition has a dual role. It can increase excitability so that they are able to respond to the environment in an exaggerated way, but it also acts as a timer because they turn down and become normal over time. So let's talk about another feature that I mentioned to you before about the different environmental events that affect neurogenesis. So we have enrichment, exercise, as I mentioned, can increase neurogenesis, but it turns out learning and some negative things like epilepsy and stroke increase neurogenesis. They increase neurogenesis, but the cells that are born are aberrant. There's also a lot of things that decrease neurogenesis, the genetic changes, stress, aging, various animal models, and actually in human clinical tissue for AD and diabetes and other tissues. This is an example of enriched environment. So here's normal housing, animals in a running wheel, enrichment. And you can see here very dramatically that there's a proliferation of cells in the running cages. And this increase in running and enrichment here, for example, can result in 45 to 50,000 new neurons over a period of 45 days, and the volume of dentate increases. So this is a highly regulatable phenomenon that experience can regulate. Here's a picture of a mouse on a running treadmill. This is actually a manufactured picture, but they usually run in running wheels. I have these superimposed for a little humor. But the truth is that this is the distance, and this is the number of granular cells, and there's a good correlation between the two. Interestingly, there's a decrease in neurogenesis with aging, dramatic. Here's that 6 months versus 21 months in a rat. The number of cells are born decreases. We asked whether or not in old animals increases in neurogenesis could be activated by running. So the animals were given BRDU and then ran in a running wheel, and they were tested in a water maze when they're 19 months of age. And what we see is that the old runners in a sedentary situation had a hard time finding the maze. But on this side, they can see the maze quite well, right there, as opposed to this sort of not really knowing where the maze is, even after a lot of training. And this is documented here, showing that the aged animals that that had a running experience, and this is these guys here, the circles, they performed in latency nearly as well as the young controls. So not only are we seeing an increase in neurogenesis, but we're seeing an increase in performance in the aged animals. Here's their quantitation on the performance. This is the young animals that are running. They're doing really well. Here's the young animals. And what we get is a significant increase in the performance of old animals that had some running experience in their ability to remember it. If we look anatomically, in old and young, we confirm that there is a decrease, but there is a significant increase. So here's the old guys. When we label BRDU, running can bring them up to the level of young that were not running. It doesn't bring them up to the level that the young animals have when they're running, but it does increase significantly the number of BRD-labeled cells. And if we look at the GFP labeling from retroviruses, these cells that are born in the old cell brain are mature. They have spines and they look good. So if you can get a new cell to be born, it's good. So now we're at the final stage of trying to understand if these are functionally significant, what role these new neurons may play, and there's been a lot of studies trying to figure this out. I'll summarize a few of them. And they're based on a series of computational models that have tried to map out the possible role that the dentate may play in the function that we know of as hippocampus, that we know that hippocampus plays. So the model was based on, the more recent model has been based on an older model by David Marr, who suggested, based on the density of the dentate, where they have so many neurons compared to their input in the entorhinal cortex, that it was a great location for what's called sparsification, and what he later described as pattern separation. So keeping memories distinct is one of the functions of the dentate. The process is taking complex memories and converting them into representations that are less easily confused. And this process is known as pattern separation. The model goes on to say that particularly the new neurons are involved in pattern integration, where the mature neurons are involved in pattern separation. That the timing of this new neurons leads to temporal dynamics, so you can have calendar-type memory and some specialization of these cells once they are there. So here's a practical example. When you park your car in a location at the beginning of the day, You can come out at the end of the day. If it's a big parking lot and you're not parking in the same place each time, you have to recall what you did that morning and separate that from the other mornings that you come out. And that's a typical example of pattern separation. Here's an animal version of it. This is an 8-arm maze, and these red bars correspond to these red bars here correspond to how separate the bars are. So the animal's put into the maze at this point, and then they have food at the end of the maze. On one trial, they go down to get food, and then on the second trial, they have to remember to go to the other arm. And they're taken out and put back in time. And some of them are very different from each other, and some are pretty close. And what's clear is that when you knock out neurogenesis, sham animals have a pretty high rate of learning this at this low rate, this low separation. But you knock out neurogenesis, and they have a much harder time learning it. When they're very separate from each other, both groups are okay. This will come more clear. Here's another task where an animal has to press their nose against these windows here. And when they're separated far apart like this, it's high separation. It's easy for the animal to make the discrimination between, say, seeing it here the first time and here the second time, versus when they're close together. It's harder to make that distinction. And here is showing that, again, on this second task, that if you knock out neurogenesis, they take many more trials to criteria than do the sham animals. But when it's very easy to discriminate, they have an easier time to do it. Henrietta and Prague went on to show that if you run the animals, let them run in a running wheel, that you actually enhance their ability to make discrimination of the small differences. So both knocking down and overexpressing neurogenesis has a reciprocal effect on behavior. This type of pattern separation has been tested in humans too, and with a very, very similar result. There's a paper by Craig Stark in Science in 2008. So a lot of people have published work on this idea of neurogenesis and pattern separation, and all coming up with pretty much the same conclusion. So we're going to let's test this a little bit. I'm going to show you one more test toward the end here. And here we have an example of a mouse that has a nestin driving TK, thymidine kinase, and you give encyclovir to these cells that are dividing, and they'll die. So it's a selective way of knocking out just the dividing cells in the dentate or in the brain, in cells that are nestin positive. So you have a way to localize that effect. And when you do that, you can show that with repeated testing with encyclovir, you can knock out about 50% of these double coordinate positive newborn neurons. But it comes back with time. So if you stop giving the drug, then the neurons pick up again. So here we are at 9 weeks later, and you can see the recovery. So this is a test we've done to look for pattern separation in another sense. So the animal's exposed to a chamber here. They're visualizing. You wait 24 hours, put them back in that same box, shock them, and then you wait for a period of time and either expose them to that box or to a slightly different box. And so here's the pattern separation. They're very similar boxes. And what happens is normal animals can make this discrimination, though it's a little tough for them. They normally freeze about 40 to 50 seconds, and then if they're shown the related box, they can tell the difference statistically, but it's tough. If you knock out neurogenesis, they can't tell at all. If you wait 9 weeks, it comes back again, so they can actually recover neurogenesis and recover their ability to make these pattern separations. This is just to show that, in fact, it's an effect that is dependent upon it being a closely related box. If it's a very different box, so this here would be the very different from this box, then they have no trouble. even without neurogenesis. So neurogenesis really is important only for discriminating between closely related objects. So 4-week-old hyperexcitable neuron neurons are not mature neurons and are required for pattern separation. New neurons are only required for discriminating between small differences between contexts. And let me skip this and come to the conclusion and say here that the brain is the organ that controls behavior. New neurons continue to be made throughout life. The ability to make new neurons is dependent upon behavior and experience. We learned about running and exercise and experience, so it's dependent on those kinds of things. Your behavior can thus affect the structure of the brain, which controls behavior. Thus, learning, music instrument, or deeply experience a new event can activate new neurons, and this can help you appreciate the finer details that exist in life. So in the last 20 years, a lot of progress in adult neurogenesis, much more to be learned of mechanisms, and new technologies are needed to visualize and see these in the future. So I'd like to stop here and thank you for your questions. I'm going to go over here to the Q&A part. Let's see here. I have a couple of questions. In one of your articles, you've written that new neurons are act differently than old ones. Newborn neurons fires up at any signal. I'd like to know that, in that case, how the memory is stored or replaced. In that case, to actually, I think it's, so they are differential. I'll just try to respond to that question. The new neurons and old neurons do respond differently. The new neurons, when they're 4-week-olds, they're hyperexcitable. They have a lower threshold for firing. They have less inhibition. And the older ones are highly inhibited and only sparsely respond. I'd like to know that in that case, how the memory is stored. So we don't really know what the source of the engram is or how information is stored within a particular cell, but the idea is that excitable moment, hyperexcitable moment, makes some change in that cell that once it's mature, it's maintained. And that is a wonderful question of how is information stored within the brain, within an individual cell. And I think there's probably some molecular event that occurred. Let's see, I'm getting a question mark here. I see. So anyway, does a link from experience to the innate immune system and the creation of olfactory receptors extend to the creation of new neurons? That's a great question. There is a significant amount of evidence now suggesting that the immune system is regulating to some extent neurogenesis. In particular, the endogenous immune system is reflected in the microglia within the brain, and those microglia are definitely involved in regulating adult neurogenesis. Now, whether or not that's involved in the olfactory receptor gene creation, I'm not sure about that. I know mostly about I don't know about that, or if anybody's done that, but I would not be surprised. SOX2 cells can differentiate into oligodendrocytes as well, right? They can, but in vivo they rarely do differentiate into oligodendrocytes. You don't see oligodendrocyte differentiation in vivo very often. In vitro, you can force the conditions to make them oligodendrocytes, but not in vivo. Let's see, Jasperger shown that ASCII SOX2 expression causes neural stem cell differentiation into oligodendrocytes. So at the time, so the point, so oligodendrocytes too for myelinating neurons. It's true that, this refers back to the previous question by, the same person, I see, okay. Yeah, so if you force ASCII or MASH1 in the new neurons, you can get them to express it, but it depends on the concentration that you're using, but normally it doesn't occur, but you can force it. That's a good point, thanks for reminding me. Professor, if newly born neurons, this is from G. Lee, if newly born neurons compete for pre and post synapses, does this mean that older cells have apoptosed? You know, you have to think about this at the synapse level. Just that every neuron's got thousands and thousands, at 5,000, it's expected 5,000 synapses on each granular neuron. So the competition is just at that individual level. So gaining or losing a few synapses may not result in death. On the other hand, if a cell loses enough synapses, it will occur. And we do see some apoptosis occurring with age, but it's not a dominant feature. It's not dominant. It's a regulatable one. Hello, would it be possible for you to post a list of papers that you referenced through this lecture in the future? Well, certainly look into that, Shane. You know, to your point, there's we wrote a review. We've written several reviews about this, and one of them in Physiological Reviews in 2014 is a good summary of it's a very, very long chapter with hundreds of references. Aha, it's Dave again. As you said, increased activity, increased neurogenesis. Why is it like more exercise means higher mitochondrial? So what people think about the reasons why neurogenesis increases with exercise is because two things. One is it's serotonin mediates this to some extent. Insulin, IGF-1's involved in it, and theta activity in the hippocampus is involved. So we know several regulators. We don't know all the details, but there are endogenous mechanisms that are affected by exercise, which affect neurogenesis, and we think that they are mediating. It's not, you know, it's not undeniable, or let's say it's not wrong to implicate mitochondria. I'm sure mitochondria are playing a role in this as well. Still, much work needs to be done. How do you relate the brain plasticity with learning? What kind of role has microglia in brain plasticity? Okay, how do you relate the brain plasticity with learning? Well, you know, if adult neurogenesis is a form of brain plasticity, which it certainly is, and we believe that these cells are important for learning information, particularly about, as I said, things that are closely related to each other. And microglia are playing a pretty strong role in particularly, we think there's evidence that they're playing a role in making these new synapses by gobbling up and phagocytizing the existing synapses on the surface of the cell. So yeah, microglia play a role. What foods, spices, oh, good. We're dropping things out pretty quickly here. How do you relate? Oh, I'm losing questions. How much time can elapse before synapse rebuilding related to recovery happens? Let me read that again. How much time can elapse before synapse rebuilding related to recovery happens? You know, that's going to depend on the circumstances. Looks like my time is up. I hope you've enjoyed this. Thanks very much. Bye.